Our dignitaries have joined, and by their permission, I will start this program.
You can start.
Thank you, ma'am. Honorable Vice Chancellor, Ranchi University, Ranchi, Dr. Kamini Kumar, respected Principal, Nirmala College, Dr. Sister Jyoti, respected Principal, P.K. Roy Memorial College, Dhanbad, Dr. B.K. Sinha, respected seniors, colleagues, and my dear students, a very warm good afternoon. On behalf of the Zoology Department of Nirmala College and ICON, I, Dr. Emma Seraphim, welcome you all in this August online gathering. Hearty welcome once again. Today, two events are set to unfold: the release of a book and a webinar on the topic collection, identification, and preservation of insects. Before we begin the day's event, let us have the ceremony of lighting of the lamp. It is rightly said, hope is like the sun. If you only believe in it when you see it, you will never make it through the night. So, let the light of lamp invite the presence of God to guide us in these darker times. May God's blessing always be upon us. So may I request Aman sir to kindly share the screen for lighting of lamp. Now, may I request Principal Nirmala College Ranchi, Dr. Sister Jyoti, to welcome the gathering and shower the event with her blessings. Over to our Principal, Sister Principal, Dr. Sister Jyoti, for her welcome address.
Thank you, Dr. Emma. Honorable Vice Chancellor, Professor Kamini Kumar, Ranchi University, Ranchi, Chief Patron of today's webinar, respected Vice Principal, Sister Shobha, respected Dr. S.K. Sinha, Dr. M.R. Rani, the organizers, and all faculty members, and my dear students, a very warm good morning, good afternoon to you all. I take this opportunity to welcome to this virtual Zoom meeting for this webinar and book release. It is indeed our proud privilege that Honorable Vice Chancellor is with us through this virtual platform to witness this one-day webinar as well as the book release. I wholeheartedly welcome you, Madam, and I express our deep regards and respects for your valuable presence amidst us. I want to highlight that you have been always with us in our journey to the search of knowledge and also in search of excellence, including our NAC assessment process, and indeed your blessing that has blessed us with grade A. We are proud of you, ma'am, and we acknowledge your valuable presence amidst us. Thank you, ma'am, for being with us, and welcome to this webinar. I highly feel privileged and proud to witness this one-day webinar organized by Department of Zoology, Nirmala College, Ranchi, affiliated to Ranchi University, Ranchi, a college with potential for excellence, accredited by NIC with grade A, in collaboration with the Association of Indian Conservationists of Nature, that is ICON, on the topic insects, collection, identification, and preservation. It's indeed a great initiative, and I feel it will be very interesting. We know that the earth is blessed with diverse flora and fauna. The divergence and vividity of the living forms create a beautiful environment, as we know. Although each form has its own contribution, but insects are a common species known to each one of us. It's because of its form, the most abundant living beings on the planet. They have beneficial role in the natural ecosystem and provide useful services to the mankind, the entire human community, I believe. I am sure this webinar will help all our students as well as faculty members to develop some more insights on the diversity and ecological role of insects. It would also suffice their academic understanding on the role of insects in maintaining ecosystem. We wish you all the best and have a pleasant time for this interaction as well as your endeavors. I hope this webinar will help you to achieve the goal of learning and gaining knowledge and enrich your knowledge bank. May God bless you all. Have a pleasant time. Thank you.
Thank you, sister. Thank you very much. Books are like the eyes of the human world. They absorb that is around and reflect the same. They envisage the face of the society like a healing mirror to which many have added over time immemorial. Today, we add to this epic a small piece that reflects the struggle of human society through the COVID 19 pandemic. Authored and edited by Dr. Emarani Seraphim, the book Quam Vita Life Beyond is a compilation of discussion and deliberations presented at 2 national webinars by renowned resource persons and learned participants. May I request our chief patron and chief guest of the event, Honorable Vice Chancellor, Ranchi University, Ranchi, Dr. Kamini Kumar, patron and principal of the event, Dr. Sister Jyoti, and esteemed resource person, Dr. B.K. Sinha, to release the book.
Congratulations.
Thank you, ma'am.
Congratulations, Emma.
Thank you, sister. Thank you, sir. Thank you very much.
Congratulations.
This is indeed the happiest moment of my life. Thank you, ma'am. Thank you, sister, once again. Thank you, sir. The 20 chapters of this book are divided into 5 sections: Nativitas, Heal, Transience, Reincarnate, and Transcendence, compiling the voyage of our world through the perilous sea of COVID-19. We hope This calamity will soon find a conclusion, and this book will render witness of what came and what went by for the generations to come. I would like to invite Dr. Sister Jyoti, Principal Nirmala College Ranchi, to speak a few words about the book. Please, Sister.
Yes, Emma. First of all, my heartiest congratulations to Dr. Emma Rani, the author of this book, that is Quam Vita, meaning life beyond. I think all of us will agree with me that the COVID-19 pandemic is a mega tragedy that unfolds across the globe. This book is titled, as I read, Quam Vita, meaning life beyond. Pages of this volume are like clear water that reflects not only the present tragedy, but also the trauma that shall ensure in the near future. This book explores the various and far-reaching aspects of coronavirus disease 2019. As you said, this book has been divided into 5 sections, and each section speaks about its origin and its effect on peace. I am sure is going to help the entire human society. So wish you all the best, and my congratulations, best wishes to you and for all your endeavors.
Thank you, sister. Thank you very much. You have been constant support and source of inspiration behind this endeavor. Thank you very much. With great joy and love, I would like to invite our Honorable Vice Chancellor, Ranchi University, Ranchi, Dr. Kamini Kumar, to inspire us with her valuable message and to announce the inauguration of webinar. Over to Vice Chancellor, ma'am.
Okay. Thank you, Emma.
Ma'am.
Good afternoon to all of you. First of all, I welcome you all on the occasion of webinar on insect collection, identification, and preservation, organized by Department of Zoology, Nirmala College, Ranchi, in collaboration with Association of Indian Conservationists of Nature. I inaugurate your webinar. Today's patron, Dr. Sister Jyoti, Principal, Nirmala College, resource person, Dr. B.K. Sinha, convener of webinar, Dr. Emma R., and author of the book. First of all, I would like to thank the organizers for organizing the webinar on such an important topic. And I would like to congratulate the author, Dr. Emma, for taking pains in publishing the book. A big congratulations to you. Insects and environment is a very nice topic. Insects are environment friendly, and we all should know about the life cycle of insects because they very well balance the environment. Early in the school times, we used to study a subject, biology, together with botany and zoology. And in that biology subject, the insect biology is collection of specimens. It is important for taxonomic research, ecological studies, bioassessment, and biomonitoring, and physiological and genetical study. Living arthropods exhibit collections which are important for educating public about biodiversity and ecological importance of arthropods. They are important source for conservation and recovery of endangered and threatened species. Now, as a student, one should know about the basic equipments such as aerial net, beating net, aquatic net, malaise, trap, light and light lamps, pitfall, and other important instruments such as aspirator, pinning insect specimens, placing insect in card points, and infrastructure of spreading butterflies. So whenever we see the insects, a person who is not a biologist becomes feared. So it is very necessary for the public to know about collection and preservation of insects, because during the exercise of collecting insects, collectors learn about relationships between insects and their environment, the importance of habitat, keys to species survival, and relationship between species groups such as host, predators, and parasites. There are many reasons for collecting insects. Insects are minute living creatures, and their identification is very because on the basis of identification, taxonomy can be developed. And when we properly store and preserve the insects, they can be studied for hundreds of years. Insects are the most common form of wildlife, and during their collection, their relationship between environment is studied. So when a particular insect you collect in a particular environment, then you can identify that this insect is collected in summer, winter, or it is collected during rainy season. It provides knowledge for tolerance, and fear is finished. So when you study the nature of the insect, obviously you can study the tolerance level, and automatically the fear is finished. You see, many insects are very beautiful, such as butterflies. And butterflies and many insects, they are responsible for pollination. When you see there are beautiful flowers in the garden or on the trees, the insect come, they pollinate the flower, and fertilization is done while they are playing on the flower, and ultimately a fruit is formed. So insects are very important in the field of agriculture and forestry. And insect study is very important because insect you study the pest also, and understanding insects make you a better person. They are directly beneficial to humans because of providing honey, wax, and indirectly they are important pollinators of crops, natural enemies of pests, scavengers, and food of other creatures. So the insects, they also kill the pest and the crop is saved. Entomological collections help scientists to rapidly identify invasive pests that affect agriculture, forestry, and human and animal health. Invasive insects and mite pests can have tremendous economic impacts. Collections also offer a lens into the past, a snapshot of the present, and a means for predicting the future, particularly with regard to how planetary biodiversity has changed and continues to change in response to global shift in climate and land use. In the villages, you see the villagers, they predict the environment with the presence of insects. Suppose a particular insect is seen during summer, then they will predict that this time there will be heavy rain, and they will predict this insect has come, so there will be no rain. These insect collections are critical for educating the public about biodiversity and the ecological importance of insects. Now, when you are having this webinar and you are going to have a nice talk from BK Sinha, so you should discuss about new policies which should be framed for employment of additional well-trained collection staff, more training opportunities for existing staff, improved facilities and infrastructure, development of new analytical methods. There are many statistical techniques by which you can identify or you can go through detailed research process. Technological advances that further enhances the ability to gain new knowledge should be should come out through this webinar, through the study of specimens. How will you improve the funding? How the increased public awareness regarding collection and preservation is to be made. So by this webinar, you should see that the public should have awareness regarding insect collection and preservation. And this will be science and society as a whole. You should initiate interest in researchers, teachers, administrators, marketing representatives, students, and those people which have a hobby to collect insects. Now, at last, I would like to thank all of you for inviting me in this webinar Zoom meeting. I know and I am confident that you all will gain a wide knowledge from Dr. B.K. Sinha, who is a very good teacher, a very good administrator, and he will give you nice lecture on this topic. I wish all success for this important webinar, which will be very useful to students, teachers in gaining knowledge. At last, I thank the author, Dr. Emma R. Serafim, for the book she has written, Quam Vitae. This is a very good book, and I am confident that this book will be very useful to students of zoology, environment biology, and biodiversity. I wish all the best and success for this webinar. Thank you so much.
Thank you, ma'am. Thank you very much for these words of wisdom. Although you are so burdened with activities, still you are so close to the subject. We feel proud to you, ma'am. Thank you very much, ma'am. Now, I request Dr. Anjali Smita to introduce our esteemed resource person. Over to Dr. Anjali Smita.
Thank you, ma'am. Good afternoon once again. I welcome all the dignitaries to this webinar. My heartiest congratulations to Dr. Emma Serafim for release of her book entitled Quam Vitae. Dr. B.K. Sinha, a learned and scholastic person who in a short span of time has made a step mark in ecological modeling and research. He has been consistently working to devise newer methods of replenishing the greenery and energy balance of our planet. His scientific thoughts have inspired teachers as well as students for working on various aspects of zoology and life sciences. Sir has huge academic teaching and administrative experiences. Presently, he is working under the capacity of being the principal of PK Roy Memorial College, Dhanbad, and I'm proud to announce that he is the chairperson of the ICON, that is the collaborated unit with which we have been conducting this webinar. His specialization is in the area of entomology, soil arthropods, biodiversity, ecology, and function. He has several fellowships to his name and has been the resource person in numerous invited lectures. So you have been to countries like Australia, USA, and you have shared your deliberations with great pride. He has guided OPHDs to are in working stage. He has edited book chapters and has several popular research articles to his name. He has been awarded Dr. Balsare Award, Gold Medal and Citation in recognition of contribution made in the field of aerosphere ecology to 2012. So we are really proud to have you with us and we wholeheartedly welcome you to this platform. Moving on, we are really pleased to have our Honorable Vice Chancellor, Dr. Kamini Kumar with us. Ma'am, your presence itself fills the whole ambience with vibrancy and positivity. Your life is a message which speaks volumes about the importance of discipline and quality control. You personify the women empowerment with great ease and poise. Your support motivates us to achieve more and more. We extend, we extend our acknowledgement to your presence and wide support in all our efforts. And we'll surely remember, ma'am, Science and Society, Nirmala College will try to imbibe all your messages on all your goodwill in doing whatever we do. Thank you so much, ma'am. So, with Emma, ma'am, shall I start the webinar?
Yes, please ask Sir to
Yes,
start the webinar, please.
So moving on towards the webinar. The webinar today involves the insects as the central theme. Insects are significant in natural ecosystems as well as they form an important part of our daily lives. It's also a wonderful research tool for many studies and analysis. Let us hear all about its story, collection, preservation, identification, being a tool, and several other things from the eminent expert who is none other than Dr. B. Sinha, Principal PK Roy Memorial College, Dhanbad. Sir, I please invite you for the lecture.
Thank you, Anjali, Dr. Anjali. Really, I'm proud to be here and I'm thankful to all of you. Firstly, my voice is clear. Can you hear me? Because my signals are poor. Internet signal is poor.
Sir, your voice is very very feeble, sir.
Very feeble, no?
It is little weak, sir.
Sir, please put that mouthpiece near your mouth so that it can be audible.
Yes.
Okay, I will try because the signal is poor. I will try to be consistent. Right. I'm using both of the systems. Two systems are being used. So I firstly, Honorable Vice Chancellor, Dr. Kamini Kumar, Ranchi University, Ranchi. She has taken out time for this webinar, which is being organized by the Nirmala College and the ICON. I thank Dr. Sister Jyoti for giving me this opportunity, and Dr. Emma, who has been consistently asking me for
Sir, sorry to
some of the other
interrupt
time.
you, but
Yes, sir, please.
yes, sir. Now it's clear. Now it's clear, sir.
Yeah, you will have to bear with because my signals are poor.
No,
Internet
sir.
signals are poor.
Okay, okay. No, sir. Now it's
Okay.
clear because. Okay, sir. Okay, okay.
I will try my best to give you the best possible because I have observed for the last 3 to 4 hours. I'm trying to get the best signal, but I will
No,
try
sir.
my
Now it's
best to
clear. Now it's clear.
get you the best out of the resources available just now, isn't it? So bear with me a little bit. So I thank you all. Thank you all of the students, those who are here to hear me. The words that has been spoken for me is very loud. I'm just a student of zoology, still learning many of the things, and I think I will be learning again today with many of you people. While talking about the things, uh, Honorable Vice Chancellor has said many of the things and has got high expectations from this webinar. She was talking about the tools that we are going to learn, employability, technology, research, and many of the things. Obviously, uh, the administrator or the academician turned administrator has got many things to give to their students in the university. When there is a focus on skill development programs, any of the area that we are going to learn will have to develop some sort of skill. Simply getting a degree nowadays is not sufficient. Degrees are no doubt important because they are the passports, but without visa you cannot enter into any of the country. Similarly over here also, if you are having the degree and you are not having the skill developed in yourself, you will be not able to qualify in any of the job. So it is very important nowadays. The second important thing is that one has to learn how to develop skill while studying. When you are in a course, you are a student of zoology, there are ample scope because what I have learned in my uh, days when I was student, I learned that we, the student of zoology, do not know the animals, those who are in and around us. Even we do not bother their scientific name even. So that starts from very much in your surrounding itself. So you'll have to learn your surrounding first. Knowing zoology means knowing yourself, knowing environment. So is the case with any other animal also. And I tell you, one has to understand this one. Human beings are not in isolation. They are a small component of the whole of the ecosystem, whole of the living world. And the, each component of the ecosystem, you know that there is a flow of energy, one, and the flow of energy is via the food chain. Actually, food chain doesn't exist, but there is a food web as a whole. So intricate mess of the food web. In that food web, we are a small component, and many of the animals are there, many of the plants are there, primary producers, primary consumers, secondary consumers, tertiary consumers, and so on and so forth. The system is living, dynamic, and that's why everything is moving. There is a life cycle of the system itself. So, learning is very important, and in the learning, we are supposed to develop many skills, even right from the development of learning skills, and especially when it comes to zoology, we have got ample field or area where we can learn many of the things. Right from preparing of the slides, preserving the animals, and even the smallest thing I tell you, when we prepare a record of the experiments that we perform in the laboratory, why we do prepare that document, what we learn out of it, that is important actually. How to label it, how, if a complete animal is there, then where should be the classification? If it is not complete animal, then what it should be, in what way it should be presented, and so on and so forth. So likewise, many of the things are there. So while giving me this opportunity to discuss in this webinar, I will be focusing a little bit of things that I have learned and I would like to share with you people. So firstly, I must congratulate Emma once again for her book on Quam Vaita.
Thank you, sir.
She has written this book during this pandemic, and a great effort she has put in over there. So it's an achievement for the Department of Zoology of Nirmala College as well as the college as a whole, and giving me an opportunity on this occasion, on this release of the book. I feel proud of it myself. That icon is also associated with it. Now I'd like to share some of the slides. Through slides, I would like to discuss things. Is it the slide is visible?
Yes, sir. It is visible.
Slide is all right. Okay. Thank
Yes.
you. Thank you.
Yes.
So just we will be starting the thing today for the today's webinar. Actually, you see, we have got three objectives here. What is insect has been talked in the very beginning by the Honorable Vice Chancellor, but still, where to collect and how to preserve it, and how to identify. So there will be three dimensions. Although insect is a very wide area, and it's very difficult to us, but I will try my best to give some justice to it. I will try my best. So, dear students, What is insect? You know it. It is generally called as hexapoda, the six-leg non-vertebrate or invertebrate. Generally, you call it as invertebrate, but it is a non-vertebrate as a whole, we call it. So hexapoda. Its body is divided into head, thorax, and abdomen. It has got single pair of antennae. These are the characteristic features. One pair of mandible, two pairs of maxillae, second joined to form labium. It's a very characteristic feature of it. Three pairs of walking leg, born on thorax in adult, tracheal system with segmentally arranged spiracles. Development may be direct or indirect. It depends upon the whether there or not. It's direct. So it is the largest taxonomic group competing with human being, direct confrontation. This is very important to understand. And that's why study of insect, knowing about the insects is very important. So that's why it is beneficial as well as harmful both. You can see the number wise, percentage wise, what the number of insects are on the earth. The whole of the blue area you can see that is 94.28%, that is insects on this earth, followed by all rest of the eukaryotes and other organisms on this earth. So they dominate. Many a times people do say that these insects are aliens to the earth. There is a story. Sometimes we say that when the earth was born, these insects they came to the earth and they dominated on this earth. So they are aliens. So you can find them everywhere, every nook and corner they are present. We can define this insects by generally, if I go a little bit faster over here, this is I have taken this classification from MS Money, which is very easy to understand, and that's why I have taken this one for the students. Although there are number of classification of the insects, but generally for the purpose of understanding for the students, what I feel is this one, Professor MS Mani's classification is good enough to understand, and that's why I have taken this for this today's uh, deliberation. So you will see the insects are basically in two groups, what is called as apterygotes without wings and pterygotes with wings. In apterygotes, there are four groups: Thysanura, Dipleura, Protura, and Collembola. Collembola dominates again, whereas the Dipleura, Protura, and Thysanura, they are less than and they do not dominate. Then comes the pterygotes. In the pterygotes, again, there are two divisions, Exopterygota and Endopterygota. In the Exopterygota, there are some 16 orders that you see, right from Ephemeroptera to Thyssanoptera. So you will find Odonata, Plecoptera, Gryloblatidia, Orthoptera, Phasmida, Dermaptera, and so on and so forth. Isoptera, the most common, Isoptera, Sopoptera, and so on and so forth you can get over here. In this group, rather, you will find the dominant one is Odonata, Isoptera, you generally find in and around you. Even the Phasmida, you find in and around you. Hemiptera also. These are very common and you see them with different seasons. Ants, the dragonflies, the grasshoppers, the stick insects, and so on and so forth. You find all these in and around you. Then comes Endopterygota, where you have got the Neuroptera, Mecoptera, Lepidoptera, the butterflies, Diptera, the flies, and Hymenoptera, Coleoptera, and others. Hymenoptera, wasp, bees and all that. Coleoptera, commonly the beetles, they are. Now here, what I am going to have today, the discussion, that will be basically on two of the insects. One, that will be the Collembola, rather the Aterigotes as a whole in general, and the other one, the uh, Collembola. The Pterygotes, that is the Coleopterans. Because presently I am working on it, and it will be better for you people to understand also. It will be easier also, rather. Some points you should remember. Insects are grouped in 27 orders, 4 Aterigote as we have seen just now, 23 Pterygota. Aterigote, Collembola, as I told you, better known in India, represented by about 210 species, under 86 genera. 4% of the world fauna of the total Aterigotes Collembola is in India. Then Pterygota, Coleoptera is the largest of all orders, about 15,500 species, about 4.5% of the world fauna is present over here in India. Then comes the Lepidoptera, the butterflies, moths, etc., of which 15,000 species are present in India. Another major group is Orthoptera with 1,750 species. 10% of the world fauna is present in India. So this becomes a very, very large group and obvious group of the insects that you see, other than the honeybee. Honeybee is very important, I tell you, is maintained because of the honeybee. Honeybee pollinates, as Honorable Vice Chancellor has said. If they are not there, we will not get the food. So they are very important for us. And honeybee pollinate. Due to pollination, the fruits bearing plants bear the fruit. So they are the important component of the ecosystem. So we should know the insects. What I feel, or rather I will be doing today, I will just not bore you much, but I will give you some pictures, good pictures, so that you can have insight in it and you can understand. And then you will be asking questions, then it will be better for me to make you understand things. How to collect, where to collect, and how it. I will be focusing in two of the things. One, the insects that lies beneath your foot, the 6 inches or 6 to 8 inches where you stand. So that is the Aterigotes there we find, the Aterigotes. And Collembola is most important, as I told you. They are commonly called as springtails because they jump like a spring, and that's why they are called as springtails. So you can see the morphology and as well as an anatomy of the insect. A typical insect, that's the morphology. That body is divided into head, thorax, and abdomen. The thorax is divided into pro, meso, and metathorax, whereas the abdomen has got measure. My work has been in the one is called as the area Bermo, the other one is Bundu, and focused generally majority of the time in the Ranchi area. So these are the areas where I have worked, collected, brought back to the laboratory, where we use tull green, Berlesi tull green funnels, and behavioral extraction of these insects are done. We will see the photographs later on also. So one thing is that behavioral drive of these students, because these insects either come towards the light or they go away from the light, temperature. So we drive them through temperature. So 100-degree bulb, incandescent bulbs are important. We use it in the funnel and keep the soil in that funnel collected from the uh, field. And these insects move out and they are collected in the vials where the alcohol or formalin sometimes are used. Generally, we use the 70% alcohol for wet collection of the insects. So soil insects are generally collected in the 70% alcohol or simply water. And you can see we have collected many of them. A few of them you are seeing. The one is the springtail. Here also, this is one acari, the box mite, the coleopteran larvae, you can see, the pseudoscorpion, and again the other acari you can see. So likewise, you can see many of the insects, they are filtered out and collected from the trash, and these are then again made the slides, and then we study. Again, some of the collembolans are there.
So the slides are not visible here. I cannot see the slide, just one slide. Insect collection, I think that's the first slide.
First slide. You want to see this? Is it visible?
No, I cannot see the other slides. Just the first slide is visible. And the other slides that you're changing, it is not visible.
Okay, just
yeah, to me.
Okay. Is it the same case with all?
Yes, sir.
The slides are not
Actually,
visible?
the same.
Slide condition is not there, sir.
Okay. Just bear with me. Is it visible now?
Yes, sir, now it is visible, sir.
Okay. Sorry. Is it clear now?
Yes, sir, it's clear now.
Okay. Thank you. So I'm just going to that slide where from I was talking about visual things are available, isn't it? So this is one of the springtail which was photographed by an instrument or microscope, the field microscope. Just I was talking about the anatomy of this morphology and anatomy of this insect, typical insect that is having, now you can see this one that I was talking about. I was talking about actually from this slide. You can see this. So springtails, collembola, acari, pseudoscorpions are there, which were collected from the trash, and this is one of the larvae of the caterpillar or larvae of the coleoptera. So this is pseudoscorpion. So you find every minute organisms or the insects you can find over there in the soil. Some more collembola you can see over here. And here again you can find the acari, collembola, and the coleoptera larva. Now just see and enjoy the photographs first, then I will be talking about other things. You can see this is Zygogramma bicolorata, the Coleoptera, which is called as Parthenium beetle. You might have seen in Ranchi many of the Parthenium beetle. So this is an infestation of the Parthenium beetle over here that was rather introduced to control the Parthenium. And presently we are working on the beetles actually, on the ground beetles. And that's why I have focused today on either the columbola or the ground beetles. And I will be talking about the preservation and collection, how the things are collected, and all so and so forth. Everything will be discussed. So you will find the other one is Urtica species. Then you can see Leptinotarsa. Then you have got the Coccinella. So all these are the coleopterans collected from just neighborhood of your place, that is Pitoria. All these coleopterans have been collected from Pitoria or Kanke area. Some of them you might have, we have collected from the rather Morabadi. So you can find all these insects in your area also. So these are wonderful coleopterans, and you will find these coleopterans in and around yourself. The longhorn coleoptera, very commonly found in the houses during the rains. The Orthognathus, Gynopus. So all these animals, rather coleopterans, you are going to get in and around yourself. Then you have got, it's a very unique organism that we have captured, that is Brachystylus sei. This is a very wonderful insect, rather Coleoptera, which was present in the leaf, and it behaves very peculiar, in a very peculiar manner. What I can tell you that when you touch it, it will behave like that it is dead. And if you leave it for a while, it will become alive and it will move. So it is a very interesting insect that we have collected. Brachinus, again beautiful Coleoptera, forage on the leaves. Another Luperomorpha whitey, another Coleoptera, beautiful Coleoptera you can see. This is Platymerus species, beautiful. This is appears on the tree trunk, and it is camouflage generally. The identification of this insect is based on the dots, prints. You see over here, the 4 dots are there on its wings, thorax and wings. So on the basis of that, we identify these insects. Many of you might have seen this one, Malarbus species. This is very unique species of the Coleoptera, and you will be surprised to know this Coleoptera only sits on yellow plants or yellow flowers and sucks the juice of the plant. So this is very unique in nature, and that's why its name is also Malarbus, according to its behavior of sitting on the yellow plants. These are another beautiful Coleoptera you can see. So you have seen a few of the insects or Coleoptera or the soil insects that have been collected during our research, and I have given you a picture of all these insects. Just to make you understand, all these insects are in and around you, in and around you. So you have to be very much cautious where they are, how they are behaving, what they are going to do. So gradually you will learn many of the things. Learning becomes easier also if you know about them. These are the tools. Here you can see one of the small microscope. This is a very handy microscope that we generally use in the field, and it costs very little. It is about 2,000 rupees. It is available in the market. And you can have the magnification eyepiece of 10x, 5x and 10x, whereas the objective is having 10x, that is built in, you can't change it. So with this magnification, you have got good magnification of the soil insects. We generally use for soil insect identification this microscope. Even when I was not having sophisticated instruments in my laboratory, we used to photograph from this microscope only with the digital camera. So there is nothing as such. You can do all these things very easily. Then there is an ambient temperature or humidity temperature measurement instrument. So hand lens is there, measuring tape is there, needles and holders are there. These are required things. Brushes, camel brush, different brush, they are required in the field. Notebook is required. Pencil and pencil sharpeners are required in the field for taking record of the things. So all these highlighter, all these things are required for your field work. So very small, nothing very sophisticated instruments are required for the purpose of study of the insects. If you want, you can choose any of the group that we have talked about earlier. I just want to show you, this is one of these instrument which was built by us only in the laboratory from Dhibri Bazaar of the Ranchi. So you can see there are the funnels and the lights are there, 100-watt bulbs in each of the funnel. And in the funnel, you will see at the end of the tube, there is a beaker in which test tubes are kept in the water. And the test tubes, in the test tubes, they filtered out, animals come. And what you have seen earlier, the animals, they are collected. All these ones, all these ones, these columbola, many. These are a few that you are seeing over here. All these are collected by this method. So, as you have seen that, or rather in the very beginning, Vice Chancellor, Honorable Vice Chancellor said that the collection is made by many of the ways. So there is net, different types of nets are there for aquatic purposes, for beating, for collecting the butterfly nets are there. So, but here I am only focusing on those things that is very small things you can have at home and you can collect, you can study, you can develop some idea, and you can be a good observer of the insects. You can become an entomologist in a very short span. So these are called as Berlesi Tallgreen funnels for extracting soil mesoarthropods or arthropods or microarthropods. All these insects can be extracted by these instruments. These are very simple. You can create it in your home. Sophisticated instruments are available in the market, costly ones, but these can be created at home. You can make. The insects like the coleopterans and others can be stored in this type of box. There are two types of this boxes, insect boxes. These are called as insect boxes. In insect boxes, you have the glass over, Now you can make yourself at home also. You can have the boxes of cardboard, of sari packing or anything other, and you can remove the upper cover of it, and you can put this cellophane and you can make the insect box. Just simply putting some cardboard inside it and some naphthalene ball dust along with the cotton you can put over there and you can store. Now for storage or rather of the insects, two very important things are there, that in dry method especially, if it is not wet, if it is a dry method, generally we dry the insects, but there is decoloration of the insects. So my suggestion is always there, you should collect the insects and you must take a photograph of it, then preserve it. Otherwise, the pigmentation will go gradually because you are not going to have many sophisticated uh, boxes for storing the insect. So this is my advice to you people. If you collect insects, please photograph it so that its original hue and color is maintained in the photograph. And the, when you go next time and study it, you identify it, it will be easier for you. The another important thing in the preservation is how to pin the insects, how to store the insects. How? So I have shown just Coleoptera in the abdomen area. Larger insects are generally pinned at the on the right or left shifted side with the pin. These are the insect pins. These are very long pins. The pins are identified with their numbers, and based on the numbers, we purchase it according to the size of the insect or the Coleoptera. And you can store insects in one level or two level also. So at a time you can store two insects also. So that is called as double layered storage of insects. So that depends on, that will depend upon how efficient you are, and you will be developing this gradually as you are very much acquainted with the storage, pinning, spreading of the insects and all that, because spreading generally is done for the lepidopterans, the butterflies. So for that there is a spreading table, and on that spreading table this small flap of paper is taken, and then the spreading the wings on both sides, keeping the butterfly abdomen within the cavity, and we spread it, and the flaps are pinned. So likewise their wings are uh, spread. So that is the way of uh, collecting and keeping them for the further study. So most of the insects generally we pin them in the thorax area, and dragonflies and damselflies center of the thorax, earwigs right tegmen, grasshopper, cricket, praying mantis, cockroaches right of the midline of pronotum, stick insects, metanotum, bugs, right side of the scutellum, beetles, right electron midway on the overall body. Generally, in beetles, we put it on the abdominal side. Flies, wasps, and bees, mesothorax, slightly right to the midline, butterflies and moths, center of the prothorax. So these are the areas where we can pin it and we can keep it in the insect box. So it will depend the size and types of the insect, which insect you are going to store, preserve for further studies or further research. Now, I would like to share a little bit of what you call as the uh, identification key. Now, it will be a tough for you just to go for the identification key. So I will give an idea. Then I will like to know from you people the queries, uh, what you want to know, how you want to do the research and all that, or other small projects of the insects. That will be better. So just
Thank you, sir. Thank you, sir, for a wonderful presentation.
Just binary, I would like to tell. Just a minute.
Yes, sir, sure.
Minutes.
Yes, sir, yes, sir.
2 minutes. 2 minutes.
बिल्कुल सर बिल्कुल
2 minutes more. I'm just sharing one screen of Amateur Entomologist Society screen. This is a portal developed by them, and it's a very beautiful portal. You can refer to this portal. I'm just giving you an idea of the things so that it will be very easy for you people to go through it. And whenever you are in problem or trouble, you can identify the insect through this portal. So it is a very interesting. Although there is one more important thing that I must share over here, that is nowadays there is a Google Lens, and in the Microsoft Edge there is a lens. You just click the insect and put the image on that lens. That will give you ample number of insects photograph and its name by which you can identify. So these are the keys to adult insects that I am going to share over here just now. So the key also refers to some groups, Protura, Diplura, Collembola, and all these ones that we have seen over there. In the beginning, I have just discussed things, isn't it? Here, why I am showing you, this is very important. Just see, key to insect orders. Start a question number 1 and follow the links until you identify your insect. Insect has wings, then go to 2. Insect wingless or with poorly developed vestigial wings, go to 29. So this is go to 2 means pterygod, and go to 29 means a pterygods. So if I go to 2, say for example I am clicking this 2, so naturally it is going to 1 pair of wings, go to 3, 2 pairs of wings, go to 7. If I am having 2 pairs of wings, 1 pair of wings, go to 3. So we will be going to 3. Body grasshopper-like with enlarged hind legs and pronotum. Extending back over abdomen is Orthoptera. So we are able to identify Orthoptera. Insect not like this, then go to 4 number. So if I go to 4 number, so we'll be having abdomen with tails. Go to 5. Abdomen without tails, go to 6. If I go to 5, so we will be having insect less than 5 mm long with relatively long antennae, with wing with only one forked wing, that is Hemiptera, and large insects with short antenna and many veins, tails long, Ephemoptera. So likewise, you have got number of keys, and through this binomial, this way you can reach to your insect, and that is a very interesting way of collecting, identifying, and finally knowing your insect. So this is the key. That is the site is being shared in this slide, so you can see that one. It's very easy. You can click it. You can put in the Google search, and you can find this site. That is this one. The another important thing is the killing jars. Very important because killing jars are important in the sense because we collect Coleoptera. For that, we generally use a bottle of 700 ml backlight jar in which we put the cotton wool and with the generally benzene. We do not use cyanides or the other dangerous materials, because earlier it was used. When we were students, we used these killing jars with the cyanides and all that. But nowadays it's not permitted. So generally we prefer to use the benzene. So benzene with cotton is there, and above that we put generally the filter paper, and then any of the insect which is being collected, we put it in the killing jar. Now, why I told you earlier in the beginning that there is decoloration. This is because of the benzene in which we put the insects. Otherwise, If it is cyanide, the coloration doesn't go. But since it is not available in the market and it will be not available to you people, so don't go for that one. And that's why killing jars are nowadays not of that quality, and we cannot go for that one also. And that's why we prefer generally the photographs. Right. So again, coming back, so all about this one, I would like to have some questions or queries from you people. Thank you very much for giving me such a good opportunity to share my things. And I have tried my best to make it so simple as far as possible, so that it is not burden and it is not making you much trouble or what you call as boring. So I tried my best to give you the things which may be in a very, very easy way. Thank you very much. Thank you.
Sir, there is a question, sir, from my side.
Yes, please. Yes,
sir, you said that we can dry the insects so that their internal organs will be dried and only exoskeleton will remain. Sir, is it shade dry or there is some method for drying them?
There are different methods for drying the different insects.
Okay,
if you go for the Lepidoptera, it has got different way.
Okay.
If you go for Coleoptera, it is a different way. If you go for the what you call as soil insects, it is a different way.
Okay.
So if you go for the soil insects, then you will have to prepare it a slide, and for that you will have to use the other chemicals. You will have to take it out from the alcohol, and you will have to put it so that in the xylene, and so that it is not foggy and all that. So the process will be totally different. Whereas if it is Lepidoptera, generally we dry it in air. We put it in the air and then we dry it. When it comes to Coleoptera, it is not required as such. It is in that fashion only. We just kill it and then we keep it in the insect box. That is the way of drying. So different insects have got different way of collection and preserving them.
Thank you, sir. I invite more questions. Yes, girls, if you have any question, please put on your queries. Unmute yourself.
Sir, a very warm good afternoon to you. I am Parul, and I'm working as an assistant professor in Lalit Narayan Mithila University, Department of Zoology. So when you were talking about the beetles, I was wondering if these beetles could be cultured in lab, like they can be bred, and like you were talking about preservation, but if we want to study some cellular aspects, means we are breeding them and we are studying evolutionary aspects, whether they can two species can interbreed or not. So is it possible to breed or culture beetles in lab?
Yes, they can be. But you see, the beetles are, because you see, I haven't cultured them, tell you very frankly, because my laboratory has been the nature itself. And I have been working on, majority of the time, I have been working on the ecology and biology. So, so I have never tried myself as a, that they call beetles being cultured, but they can be cultured. It's not that we will have to provide them with their host plants. Say, for example, the Parthenium beetle. You can culture them. Easily you can culture them. You can keep it in the laboratory. You will have to provide them with the Parthenium. You can culture them. You will have to provide their foraging plant.
Okay, sir, I think I should try this out. This was very interesting.
Right.
Okay. Thank you, sir. I think I'll try this out, sir. This was very, very interesting for me.
Sir, even I was amazed to know this Parthenium beetle, which was introduced to check the growth of Parthenium, which has become a nuisance. and causes allergy during the springtime because of, yes, it's this grains, pollen grains. Yes, sir.
Right.
It was very nice, sir.
We are going to publish one paper on this Parthenium beetle in Ranchi, actually.
Okay, sir.
Shortly we will be having we have completed their research on this Parthenium beetle. And
Okay.
in the Kanke area, the domination of this Parthenium beetle is very much.
I see. Yes, sir. Parthenium is actually, yes.
There is very heavy infestation of the Parthenium.
Heavy, very heavy, sir.
Agriculture college people have brought the seeds. Along with the seeds, the Parthenium seeds are also there from Mexico.
Yes.
So likewise, the Parthenium has spread over there. In
Yes.
83, this Parthenium beetle was introduced.
I
Firstly, it
see.
was said that it was gradually it became a nuisance in the Parthenium beetle also. But later on, it was found no, it is not doing any nuisance. There was a committee set up for that one also.
Okay.
But nowadays, it is very good biological control for the Parthenium.
Yes, yes, sir. Thank you, sir. Thank you, sir. Students, if you have any query, please. Or teachers, if you have any query.
Excuse me, ma'am.
Yes, sir.
Hello.
Yes, yes, sir. You are audible, sir. Please ask.
I just want to share information that in Loyola College, Chennai, Department of Entomology Research Department is there, and the director of there is Professor Father Ignace Muthu. And there I have been over there, and there the people are working on molecular studies, and they are also culturing and keeping all the sorts of beetles and all the sorts of different pests also. And they have got a separate department for that. And this is the added information that I want to give. Someone, perhaps Parul Banerjee was asking this. So this is the information to you.
Thank you, sir. This is precisely what I wanted to know, that whether you can do molecular research, take out some cellular
over there in Loyola College, Chennai, they are also performing all the molecular studies related to the pest and all. They are starting the DNA and going for the molecular studies.
Yes, yes. So that would be very interesting.
Nice.
Thank you.
Thank you, Ritesh Shukla, sir. Thank you very much for this information. Yes. Over to Anjali, madam.
Thank you, ma'am. So we had really a wonderful session today, and so the inputs that you have shared, I'm sure it's not only useful for students, but for we also, for teachers also. They are just valuable, and that the way you have put it out was just so interesting. We could not just move out of our seats. It was a wonderful presentation. The pictures along with it's wonderful, and I wish and hope that every one of us have properly enjoyed it. So, with the permission of Dr. Emma, shall I propose my vote of thanks?
Sure. Dr. Anjali, please.
The webinar was indeed a scholastic event for all of us, and our students have been enriched with the brilliant lecture given by Professor Dr. B.K. Sinha. I extend my sincere thanks to the Vice Chancellor for her valuable presence and boosting our morale. My valued thanks to our dear Principal, Dr. Sister Jyoti, who is the strength of success behind all our efforts. You are an inspiration for us that help us to work amongst all odd systems. My special thanks to Dr. B.K. Sinha, Principal, P.K. Roy Memorial College, Dhanbad, and Chairperson of ACON for being the chief resource person of the day and giving his deliberations in such a lucid manner. Thank you, sir, for enriching all of us. We are really obliged for this wonderful presentation.
Thank you very much.
I extend my humble gratitude to Dr. Emma Seraphim for all her efforts in conducting this webinar, without whom this webinar would not have been possible. Thank you, ma'am. Thank you for all your efforts. And again, on the behalf of everybody present here, I congratulate you for the release of your wonderful book, Quam Vita. I extend my thanks to Dr. Veena Pani and Dr. Manisha Kumari for their constant support and dedicated work. I thank Mr. Aman Kumar Jha for his logistic support. A very special thank you to all the participants, my seniors, my teachers who are present here and have been with us in this webinar. And a very, very special thanks to all our dear students who are the source of an inspiration, a motivation for we teachers. We want to work for you, we want to work with you, and I hope our efforts are definitely going to benefit you all in all aspects of your life. I thank the opportunity. I thank God for giving us this opportunity today. And at last, I thank all those who directly or indirectly have been a part of this program. May God bless us all. May all of us stay safe and healthy during this pandemic. So may I now please request all of you to arise for national anthem.
Thank you, everybody. Thank you once again. Please remain safe. Now we are ending the meeting.
This is